Welcome to today's topic on redox potential, phosphate group transfer potential and ATP. This is one of the 5 topics covered under the syllabus Bioenergetics Unit 1. Myself Dr. Mausmi Shankaradala, working as Assistant Professor in the Department of Biochemistry, SGCWA, hereby gives a brief note on what the above mentioned topic is. Learning outcomes will be on redox potential reaction and phosphate group transfers involved in. A redox reaction, also known as reduction oxidation process, involves the transfer of electrons from an electron source or donor to an electron acceptor, often referred to as an electron sink. In contrast to the electron sink, the electron source gets reduced. The reduction potential is a measurement of the propensity of molecules to pick up electrons. Redox process can also be discussed in terms of its opponent, that is the oxidation potential. However, in modern practice, we refer to reduction potential values as redox potential E0 values and employ them. These numbers have bearings of thermodynamics, K values and Gibbs free energy. What is standard reduction potential? Two half reactions of reduction can be thought of a redox reaction. A redox reaction's favorability is assessed using the potential of the reduction half reaction. The object being reduced is written to the left side of the each reduction half reaction, and the object being oxidized is written on the right hand side. For instance, the redox reaction zinc plus copper can be viewed as two reduction half reactions. Zinc plus 2 electrons gives zinc, copper plus 2 electrons gives copper. Spontaneous reactions have E0 overall always greater than 0 according to the following equation, which is then nothing but E0 is being reduced and E0 being oxidized. The reduction potential of the half reaction under the standard conditions is represented by the symbol E0 in the equation above. Keep in mind that standard circumstances are defined as follows: at pH 0, standard pressure and temperature. These circumstances are not biological. The redox potential is a measurement of the substance electron affinity or electron negativity in relation to the hydrogen, which is fixed at 0. Positive redox potentials are found in substances that are more strongly electronegative than hydrogen, that is, capable of oxidizing hydrogen. Negative redox potentials are found in substances that are less negative in hydrogen, that is, able to reduce hydrogen. Reductions and oxidations are invariably related. We refer to them as redox reactions. In a redox reaction, electrons release free energy when they move downhill. To represent this, we use the symbol delta G, which is followed by a minus sign. In a redox reaction, electrons must be forced to travel uphill by the input of free energy. We indicate this by putting a positive sign before delta G. A positive plus redox potential is attributed to any material that is more electronegative than hydrogen, whereas a negative redox potential is assigned to those that are less electronegative. The higher the redox potential difference delta E between the two compounds, stronger the spontaneous electron flow from the less positive to the more positive substance. What is the significance of biological redox potential? Applying the Gibbs free energy change equation to the redox process, reducing potential is a tendency of a system to either donate or gain electrons. Redox reactions occur when two or more chemical species exchange electrons and change how many oxidations they have. The first and the second laws of classical thermodynamics provide the basis for the Gibbs free energy change equation, which is used to determine whether a given process is thermodynamically feasible, that is, likely to occur spontaneously. Since redox reactions provide the energy needed for maintenance, development, reproduction, motility and other biological processes, it is very important to comprehend and quantify reducing potential. Two half reactions, one for the electron donor and one for the electron acceptor, can be used to conceptually split redox reactions. A redox reaction's reducing potential is influenced by the chemical species involved and their chemical activity on how close to concentrate they are diluted in the solutions. Every chemical species has a reducing potential determined by its propensity to provide or take electrons. The tendency of a solution to gain or accept electrons with regard to a reference electrode is used to calculate relative reducing potential. The hydrogen electrode is a conventional reference electrode. Water is the ultimate electron sink and oxygen the ultimate electron acceptor in aerobic metabolism. The aerobic oxidation of proteins, lipids and carbohydrates releases a significant quantity of Gibbs-free energy, ATP. As we all know, energy is necessary for all the living things including human, animals, birds, insects and plants in order for their tissues, cells and other organ systems to work correctly. As is a common knowledge, animals acquire energy from grazing on green plants, which in turn derive their energy from sunshine. Energy serves as a fuel source. Although the food we consume gives us human energy, there are other ways that energy is made and stored in our bodies. Considerable quantities of free energy cannot be stored by a live cell. Overuse of free energy could cause the cell to become hotter, which could lead to the excessive thermal motion and eventually cell destruction. Instead, a cell needs to manage that energy in a way that allows it to be stored safely and released when needed. To do this, living cells use a substance called as adenosine triphosphate, ATP. ATP is frequently referred to as energy currency of the cell because, like money, it may be used by the body to meet all of its energy requirements. But how? It works in a manner almost equal to a rechargeable battery. Energy is released when ATP is broken down, usually by removing its terminal phosphate group. The energy used by the cell to perform tasks, usually by binding the released phosphate to activate another molecule. For instance, ATP provides the energy needed to move the contractile muscle proteins during the mechanical work of muscular motion. Adenosine triphosphate or ATP is referred to as cell's energy currency. It is an organic molecule made up of sugar adenine phosphate groups. The body uses these molecules as energy for a variety of metabolic functions. It is known as energy currency of the cell. Now, in the cellular respiration, 24 electrons move down the respiratory chain to the ultimate acceptor, that is oxygen molecules for each glucose molecule that is respired. The redox potential of a carbon reduced to the degree found in carbohydrates such as glucose is roughly minus 0.42 volts. Whereas in photosynthesis also, in order to produce the glucose molecule, the redox potential of the oxygen is plus 0.2 volts, which holds 24 electrons in water molecules. These electrons must be extracted and pushed uphill to carbon atoms, where they partially reduce to carbohydrate, which has a redox potential of minus 0.42 volts. Since electrons are traveling against the gradient in this instance, that is in the form of positive to negative, rather than along it as they would during the cellular respiration, the delta G is positive. Energy must therefore be added to the system. Two half reactions, one for the electron donor and another for the electron acceptor, can be used by the conceptually split redox reactions. The chemical species participating in the redox reaction and their chemical activity roughly equivalent to the concentration is diluted solutions. Every chemical species has a reducing potential determined by its propensity to provide or take electrons. The tendency of a solution to gain or accept electrons with regards to the reference electrode is to calculate relative reducing potential. Water is the ultimate electron sink and oxygen the ultimate electron acceptor in aerobic metabolism. Proteins, lipids, carbohydrates like glucose also undergo aerobic oxidation, which releases the Gibbs free energy. A portion of the liberated Gibbs free energy can be stored in high energy phosphate bonds too, and thus the carbon molecules generated during the metabolic process helps as the electron acceptors in the fermentation, which is an intramolecular oxidation. Coming to the phosphate group transfer, transphosphorylation refers to the transfer of phosphoryl group from a phosphorylated compound from one compound to the other. How this can be related to the potential? Phosphoryl transfer potential is the standard free energy of the hydrolysis, which means comparing of the tendency of the organic molecule to transfer a phosphoryl group to an acceptor molecule. So what are the enzymes that are involved in the transferring of the phosphate? Kinases are the enzymes that facilitate phosphate group transfer to the substrate, which are greater than 500 kinases that are being predicted as far as the human proteome. And the phosphate group, when it is transferred from fructose 6-phosphate producing fructose 1,6-bisphosphate, this step is catalyzed by the enzyme phosphofructokinase. Example of the phosphate group transfer. One example is the coupling of the hexagonic adenylation of the fatty acid, okay, and also which is explained in the previous equation, along with what is the phosphate group, which is nothing but the phosphorus atom which is bound to the 4 oxygen atoms, but it has many important roles along with the sugars, bases and up for the nucleic acids. Now phosphorylation of ATP, the transfer of the phosphate from donor molecule of ADP to produce ATP is called ATP, and the phosphate on the donor molecule must have always